Hello, and today we'll be discussing oxygen binding to hemoglobin. We'll start off with a discussion on the structure of hemoglobin. We'll go over how oxygen binds specifically to that structure, to hemoglobin. And then we'll touch on a couple of components and some things that affect the affinity of the binding of oxygen to hemoglobin and how that differs in comparison to the lungs and the capillaries. And then we'll touch on how water plays a role in ridding the body of carbon dioxide ultimately. Okay, so we'll start off with the structure of hemoglobin. So hemoglobin is somewhat of a complex 4-polypeptide chain dimer. It consists of 4 individual polypeptide chains, and each contain their own binding site, which is referred to as the heme group. So it consists of 2 alpha chains here at the bottom and 2 beta chains. The dimer on the left is known as the alpha 1 beta 1 dimer, and on the right the alpha 2 beta 2 dimer. And you can see here these planar structures here labeled as heme with the iron in the center there. So these heme groups consist of not only iron but also all the different carbon atoms and the hydrogen atoms and all the bonding that creates this flat planar structure here, which is known as the protoporphyrin. And so that also plays an important role when it's time for oxygen to bind. Okay, so let's get into binding. So we'll be using these structures, or these diagrams here, these pictures, but let's focus on just the center here, this center flat-like structure and this iron atom here. We can disregard these outside labels here. We really want to focus on is this center ring here. And also here at the bottom you can see another depiction of the heme group itself here. So when oxygen binds, it binds at the heme group, specifically to the iron atom. And when originally when oxygen is not binded to hemoglobin, it's referred to as deoxyhemoglobin. And the way that structure is here is shown here is due to the density of the iron atom being so large it sits right under this flat structure of the protoporphyrin right here. And so we see that this iron is also attached to a histidine residue, which is ultimately attached to the alpha helix of one of the polypeptide chains of this hemoglobin protein. And so when oxygen binds, it attaches here to this iron, changing the electron density of iron, causing a shift in this upwards direction. And as you can see here, when oxygen is binded to the hemoglobin, it pulls that iron up because of that density change, which then pulls the histidine residue up, which ultimately pulls the entire alpha helix up, creating a significant conformational change overall in the structure. And so when this occurs, rotation in the protein results, and these changes actually affect the other heme groups throughout the protein to have a higher affinity to bind to oxygen. And so when oxygen is not bound to hemoglobin, it's known to be in its tense state, whereas when it is bound, that's its R state, or relaxed state. And it's known to have cooperative binding with oxygen due to those surface changes, those conformational changes influencing other heme groups to bind oxygen. All right, so now let's talk about the binding capacity for oxygen. So in the lungs, so as you can imagine, in the lungs, we take in a large amount of oxygen, and once that oxygen is taken in, it's then transferred and transported throughout the bloodstream, and then eventually delivered to the tissues throughout the body. And so when that occurs, the lungs have transferred all the oxygen and are now ready to accept any incoming carbon dioxide that has been produced as a byproduct after cellular respiration occurs, which is after oxygen is eventually delivered. And so that's different in comparison to the capillaries in that, well, the capillaries at that level, the red blood cells are ready to deliver oxygen because they have been oxygenated. The transfer occurs to the tissues throughout the body. And now the hemoglobin at that cellular level is ready to accept carbon dioxide to eventually deliver throughout the lungs. So as you can see here, it's not necessarily one or the other, but more of a just continuous cycle of exchange of oxygen and carbon dioxide through that binding via hemoglobin. Okay, so a couple things occur at the level during the exchange and transfer of oxygen and carbon dioxide and all the binding. A couple of things occur that affect the affinity of hemoglobin to either accept carbon dioxide or oxygen. And one of those things is known as the Bohr effect. And so what occurs with the Bohr effect is changes in pH and changes due to CO2 concentration. So as the concentration of hydrogen ions increases, that'll decrease our pH, and these histidine residues throughout the structure end up forming salt bridges due to this excess amount of ion concentration. And so along with that, salt bridges are also formed through the formation of carbamate. And so that occurs when carbon dioxide, which comes from the cells, reacts with water via carbonic anhydrase, which also in turn produces more, increases the hydrogen ion concentration. And so both of these effects decrease the affinity for hemoglobin to bind more oxygen. Because again, the goal is to bind oxygen with an affinity that's high enough to bind, but also not so high that you can't deliver the oxygen throughout the tissues in the body when it's needed. And so that stabilizes our T state. Okay. And now at the capillary level, excuse me, another incident that occurs is the Haldane effect. And so after the transfer of oxygen occurs, now you have no oxygen in your cells, and the formation of carbon dioxide as a byproduct through cellular respiration starts to occur. And so that decrease in oxygen in the hemoglobin increases the affinity for carbon dioxide to then bind to hemoglobin. And so that oxygen delivery to the cells stimulates cellular respiration. Carbon dioxide is then formed as an additional product. And again, with carbon dioxide and water, with the help of carbonic anhydrase, we form carbonic acid, which will then dissociate into more hydrogen ions and CO2. And so this is pretty much the home stretch. So that carbon dioxide is then bound to the hemoglobin and is transported eventually to the lungs. And the increase of the amount of CO2 into the lungs is what allows us to exhale and rid of that cellular respiration. Thank you very much.